Hi class, I'm Jamie Zellers-Batts and I will be teaching your course for this semester. I am going to be doing my videos just like this. I keep it real. I'm in my basement office, so sometimes you'll see a big huge mess behind me and sometimes you'll just see this boring wall, and sometimes my kids will walk in and sometimes my dog will walk in and sometimes my husband will walk in. So I'll try to be as professional as I can while still working from home. So this is our introduction, and I'm going to review some of the basics of the course as well as some of the basics of anatomy and physiology. So this video is going to include me like this, and it will also include the PowerPoint that is called the Introduction and Review PowerPoint in the week 1 or beginning folder that you'll see as a PowerPoint file. So you can follow along with that or just follow this. Some students like to print out the PowerPoint and act like they're in a normal face-to-face class and take notes right along with it. Or some students just like to take notes on their own. It's totally up to you how you choose to work your at-home independent learning. So whatever you feel like is easiest is fine by me. So here we go. I'm gonna switch to screen share mode and share my PowerPoint with you. There we go. So again, Zellers is my maiden name, and on some documents you'll see Zellers, and some documents you'll see Batts. My real name is Jamie Batts now because I'm married for like 8 years now, but it'll go back and forth, and you can email me at jzellers, and I still get all the emails, so have no fear. It's Bio 203 if this is my Harvard Community College course, or it's Bio 220 for my CCBC course. I'm teaching A&P at both institutions, so the course number might not be exactly the same. But so a little bit about me. I have my bachelor's degree from University of Maryland, both in biology and in secondary ed. I taught high school for about 8 years before I started staying home with my children, and I had been teaching at the college for a while then also, and I have a master's of life science from University of Maryland as well. It's not really my 8th year, it might be my 10th year at HCC and like my 11th or 12th year at CCBC, but I've taught pretty much any biology course they could throw at me. So you'll see my family. That's me and my husband on our wedding day and my 2 kids. My son Brady just went off to kindergarten, and my daughter Tori there will be in kindergarten next year. And so let's talk about the course and some of the things that go along with the class. My A&P or Mastering A&P is for my Harvard Community College students. MasteringAMP.com is an awesome website for those of you at CCBC. It's the Connect Ed. It's the same exact thing. It's just from a different publisher. So it has all the chapter quizzes and practice tests and e-text and interactive study tools and lots of things. And actually in this hybrid course, you will have required assignments from that website. So make sure you familiarize yourself with the syllabus and course expectations to learn about that. Same thing with lab materials. For Harvard and HCC, the lab materials are slightly different, but you will be using all of those materials, so please make sure you bring them to class. So let's talk briefly about study skills and study strategies. It's super important in a hybrid course or an online course that you are a master of time management and your own schedule. So set that schedule, stick to it, make sure that you are paying attention to where you're using your time and what you're doing while you're studying. Yeah, you spent 6 hours studying on Saturday, but were you really on Facebook for 2 of those hours? You know, block out those distractions and use your time effectively and efficiently. Make sure you're reading your books, make sure you're doing the reading guides, make sure you're doing the work that is assigned to you in a timely manner. It says here 2 to 4 hours per chapter is about what you should be using to read and get through the chapter, and that's pretty much about right. And then if you review your notes for about an hour a day, you're looking at spending somewhere between 3 to 4 hours a day on course material. And that's about right because really you're not in class for 3 hours a week with lecture. So you should be making sure that you use those hours outside of lab. Make sure you don't procrastinate. Make sure you ask me questions. And during the first couple of weeks of the course, we'll do some study skill and learning activities to help you learn how you learn. So this class is really all about memorization. So find the way that you memorize best, whether it's making a rap song about everything, writing a poem about everything, or maybe you memorize best by just writing everything down and recopying and recopying and recopying until your hand cramps. Then that's what you have to do. Make sure that you seek out tutoring. Both colleges, HCC and CCBC, offer free tutoring on campus, so make sure you get in touch with them. Actually, CCBC offers online tutoring as well. So contact the Student Success Centers and get in touch with them. Make sure that you are making friends with the people in lab so that you can get to know them and form study groups outside of class. Let's talk about some basic chemistry. This is a little review of basically chapters 2 and 3 for you so that we can make sure that we cover everything really quick, even though these things are super important for you to have learned about already. So some basic chemistry, CHNOPS, right? Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Those are the main ingredients in your body. You should know what those things are. There's also calcium for bones and membranes, as well as overall homeostasis, nerve impulses, and muscle contractions. Potassium is good for muscle and nerve contractions and impulses. Sodium, same thing, as well as blood volume. So our chloride is there for blood volume also. So sodium and chloride, right? Sodium, table salt, very, very important in balancing our overall water balance in our body. Magnesium works as an enzyme cofactor. We'll talk about that later. Iron is important in oxygen transport, and our thyroid hormones use iodine. So let's talk briefly about pH. Again, I kind of talk fast, but that's because I get excited about all this stuff. So pH is actually a measurement of the concentration of hydrogen ions, H+ ions, in a solution. It's measured on a logarithmic scale from 0 to 14, where 0 is super acidic, and 14 is very, very basic or alkaline. The pH of 7.0 would be distilled water, and that is the only neutral solution. And that is where there's a perfect balance between hydrogen ions and hydroxide ions, OH ions. Human blood is slightly alkaline, 7.35 to 7.45. Guess what happens when you go to 7.47? You don't want to know. Coma, even death. So it's super important that your body is staying within that range. So let's move on from pH and talk about carbohydrates. Carbohydrates, or polysaccharides, can be broken down into monosaccharides to form glucose. Glucose is our main energy source used for fuel in the majority of our body cells. Glycogen is the stored form of carbohydrates. That's in the liver, skeletal muscles. Glycogenesis, that's the making of glycogen from glucose. Glycogenolysis, that suffix lysis there, is the actual breaking down of glycogen to make glucose. So they're opposing forces. This chart comes from one of your textbooks, and it's just reviewing the basics of mono, di, and polysaccharides, what they're used for, and any special remarks in their body, like polysaccharides, like starches or cellulose, which are only found in plants, whereas glycogen is stored in animal cells. So we won't talk much about starch other than its nutritional aspects, right? Lipids, or fatty acids, they can be saturated or unsaturated. Unsaturated can be omega-3 or omega-4. They have very important health implications. Those are our essential fatty acids. They are a preferred fuel source for many of our tissues, including our cardiac muscle cells prefer to use fatty acids. Kind of interesting there. There are several different categories of lipids in our body. Really, what you just need to know for the basics are what a fatty acid is. And if I said the word phospholipid, you should recognize that as a lipid with a phosphate on it. Basics. Just the basics, right? Proteins are the most abundant macromolecule in the body. We're basically walking, talking bundles of protein. They contain carbon, hydrogen, oxygen, and nitrogen. They're made of the 20 different amino acids, and there are essential amino acids and non-essential amino acids. And basically what that means is essential amino acids are ones that our body cannot make. So they're ones that we have to eat or bring into our body, which is why it's important that we're eating a healthy, balanced diet so we can bring in all of those essential amino acids to create all of the proteins that we need to have healthy skin, nails, bones, heart, muscles, etc. Proteins basically do everything in our body, from acting as structural proteins to acting as our major contractile units in our muscle cells to transporting things through our body, across the cell membranes, etc., regulating our metabolism in the form of enzymes. They also work as hormones like insulin. They also work in our body's defense in the form of antibodies, and they can also be buffers, regulating pH throughout our body, which we mentioned is so super important. Take a look at the basics of enzyme action. Right, enzymes are catalysts for reactions. They take substrates like these two little guys here. They bind into what's called the active site of the enzyme, and they form a product. Some things about enzymes is those reactions can then be reversible. The same enzyme can do the reaction forward and backwards, meaning this product can be broken back down into its two substrates. Enzyme action is very dependent upon temperature or pH. If the temperature or pH range changes, then that enzyme may not work correctly. And you also have the issue of enzyme saturation. If you only have 5 amylase enzymes, you're only going to be able to break down starch as quickly as those 5 enzymes can work. So you're limited by how many enzymes there actually are for that particular substrate. Nucleic acids, the final macromolecule, they store and process our genetic information. This is the DNA and RNA in our body. DNA is deoxyribonucleic acid. These molecules are directly involved in protein synthesis, in the making of protein. So in other words, you bring in the protein from nutrition, your body takes those nutrients, takes those amino acids, breaks them down into their little subunits, and then your body, with the direction of the DNA and RNA, creates new proteins that your body needs from those amino acids that you brought into your body from eating. So kind of a big picture there. The overall structure of nucleic acids, they're made up of nucleotides. The nucleotide is what you see right here. It contains a phosphate base, a sugar backbone, and this nitrogenous base right here. The nitrogenous bases of a DNA could be A, T, C, or G, adenine, thymine, cytosine, or guanine. And in RNA, they are adenine, uracil, cytosine, and guanine. There's no thymine in RNA. It's a uracil instead. RNA is single-stranded, DNA is double-stranded. DNA never leaves the nucleus, and RNA goes out to the ribosome to make protein at the ribosome. That's the whole function of the ribosome, which we'll talk about in a little bit. ATP, adenosine triphosphate, is also a nucleotide, right? It has a sugar phosphate backbone right here, and then the nitrogenous base, adenine, right here. The energy of the ATP molecule is stored in this third phosphate group. So in other words, this phosphate group can actually pop off and float away, leaving ADP. ADP and its separate P has a lot of energy. So when that third phosphate pops off, energy is released. ATPase is the enzyme that pops that third phosphate back on. So it's actually a reverse reaction. This arrow kind of looks funky. Sorry about that. My PowerPoint must have been acting weird when I made this. So you have ADP plus P plus energy creating ATP. Then when ATP is broken down, it creates ADP plus P and energy. This is energy to blink, energy to breathe, energy for your heart to beat, energy for your, sodium potassium pump, whatever. All of the things in our body that happens that uses energy requires ATP. Just to blink, for instance, I actually looked this up. Just to blink one eye one time, okay, that's winking, right? Just to wink is like 10,000 ATP. So that means when you blink, that's about 20,000 ATP. And you blink how many times a minute, right? I mean, you're going through this ATP stuff like crazy. So you need a constant ebb and flow of this reaction right here. So let's talk about some different types of cells in our body, like the liver cell, the muscle cell, the neuron or brain cell. They're using lots of ATP in their lifetime. They're getting turned over every 5 to 6 days. new liver enzymes or new liver cells are being created. What about a muscle cell? Every 30 days, the nerve cells have a very, very long lifespan for some of their ingredients like phospholipids and cholesterol. So they need these main ingredients to maintain themselves for that long. Let's go on and talk more specifically about the cell. This is chapter 3 stuff. Cells, of course, have the inside and the outside. The outside is the extracellular, the inside is the intracellular, and then you have the plasma membrane that separates the two, right? The plasma, inside the plasma membrane is the cytoplasm, and that is all of the juice or the cytosol, as well as the organelles, all the stuff inside the cell. So let's talk about some of that stuff, right? The cytosol, the fluid itself, has all the nutrients, the waste products. It's typically high in protein, high in carbohydrate, low in fat, and those organelles have specific functions. They're all in the chart of your books. I'm not going to read through it because you should know it already. This is all review. But yes, you should know the functions of centrosomes and cilia and ribosomes and mitochondria and rough versus smooth ER and the nucleus. Let's talk about mitochondria. Mitochondria have a very specific important job, and that's the role of aerobic metabolism. Anaerobic metabolism takes place in the cytosol. The aerobic metabolism, or cellular respiration, takes place inside the mitochondria, where glucose and oxygen combine to create carbon dioxide and water and ATP. The first process is glycolysis. This happens in the cytosol. That's where glucose is converted to pyruvic acid, and there's a net gain of 2 ATP. So if there's no oxygen available, all of that pyruvic acid gets converted into lactic acid, and that's when you feel the burn, right? That's when you're walking up those 10 flights of stairs because the elevator broke and your thighs are burning. There's also a little transition reaction if there is oxygen present, where pyruvic acid is converted into acetyl-CoA, and then that acetyl-CoA is reduced by a bunch of coenzymes in what's called the citric acid cycle or tricarboxylic acid cycle or the Krebs cycle. It just depends on what book you read. It's all the same thing. It's a cycle, and it is right after that pyruvic acid has been converted into acetyl-CoA. Carbon dioxide is a waste product from that reaction, and lots of little coenzymes are released which go into this electron transport chain where lots and lots of ATP is then made. We're going to get all the super duper details of this, and yes, you will know all the ins and outs, even if you kind of faked it in all the prerequisite classes, because I know it's tough. You're going to get all the ins and outs of this in A&P 2, but just know the big picture stuff for now. Nucleus, that's where the DNA is. I kind of have a little pet peeve with the nucleus. A lot of people say the nucleus is the control center for the cell. In a high school biology class, that would be correct, but really it's not the nucleus that controls the cell. It's the DNA inside the nucleus that controls the cell. So just make sure you pay attention to that. Inside that nucleus, like I said, is DNA, and only when the cell's getting ready to divide is it formed in these nice little chromosome structures. Chromosomes, which are highly coiled, highly folded, basic little knots of loose stringy DNA called chromatin. They have the bases, ATCG that I talked about, and that triplet code, which some of you may recall, right? 3 bases is the code for one specific amino acid. There is a process called cell differentiation that you need to be familiar with, and that's just the idea that as the body is growing in utero from an embryo, the cells are basically deciding to be different types of cells. Fat cell, a liver cell, a stomach cell, a skin cell, a nerve cell, a muscle cell, etc. And that is done by the turning on or off of specific genes. So in other words, a liver cell, a stomach cell, a skin cell all have the same DNA inside of them. It's just which genes are being expressed that make them different. So that's kind of an interesting fact that a lot of students don't quite realize. My master's degree is actually in genetics and molecular biology, so that's more of my thing than A&P, but I've taught A&P for forever, so it's kind of become my thing. Let's talk about protein synthesis, and I'll be trying to be quick and general about it, even though, like I said, I'm this is kind of my thing. The role of gene activation is what protein synthesis is all about. So you have these genes that are just sitting there waiting to be turned on, waiting to be told to create the protein that they're designed to make. So the DNA stores all the genetic instructions for making the proteins, but really it's the protein that's going to determine what that cell is and what its function is. So there's transcription and translation, and then there's processing. Transcription is when that DNA is made into RNA, and the translation is then when the ribosome takes the mRNA and creates that amino acid chain. Let's move on to talk about the plasma membrane and its role in being selectively permeable. It regulates the exchange, it monitors the environment. It can also help to structure, anchor the structure to surrounding cells or surrounding membranes. The plasma membrane allows certain nutrients to come in and certain waste products to get out, so it has to be selectively permeable for that to happen. Sometimes it is impermeable, and sometimes it's freely permeable, depending on where in the cell's life it is. Overall, it's mostly selectively permeable, and it's going to restrict materials based on size, shape, electrical charge, as well as solubility. Because it's a phospholipid bilayer, it doesn't just let any old thing through. It's going to repel some ingredients. Diffusion is a function of the concentration gradient. So solutes are going to move down that concentration gradient until blank is reached. What has to be reached in order for those solutes to stop moving? It's a word, it's a long word, and it begins with an E. Are you saying equilibrium? Hopefully. So solutes move down their concentration gradient until equilibrium is reached. There are several factors that affect how quickly that's going to happen. First is the distance that that particle has to move. How thick is that membrane? How big is that molecule? How warm is it, or how cold is it, right? The warmer it is, the faster those molecules are moving, the faster diffusion is going to happen. How big is the gradient? If the gradient is just, a 53% to a 47%, it's not going to take that long. Whereas if the gradient is a 97% to a 16%, it's going to take a lot longer, and it's going to happen. The molecules are going to move faster because there's a greater distance or a greater difference, but it's still going to take a longer amount of time. And electrical forces, electrical charges can facilitate that process much quicker. The tonicity is how that cell will respond to its environment. A hypotonic solution is going to cause a cell to gain water. It could cause the hemolysis of red blood cells. Those red blood cells will actually rupture. In a hypertonic solution, the cells are going to crenate or get shrivelly and wrinkly and basically lose all of their water to the surrounding solution. So this is what's happening when you overhydrate. When you overhydrate, you're creating this hypotonic solution where the cells then start drinking up lots and lots of water to try to create equilibrium, and then they burst. Whereas when you're really, really dehydrated, your cells could be shrinking and shriveling up. So carriers and vesicles, these are all little parts of the cell membrane. They're specific. They have saturation limits. They can be regulated by other factors. There's co-transport or counter-transport, meaning bringing them in together or bringing one in and one out opposite of one another. The sodium-potassium pump is a really good example of counter-transport. There's also active transport, which is specifically using ATP energy to pump something across the membrane, an exchange pump like the sodium-potassium pump. So we look at the sodium-potassium pump in specific, and we see that every time it pumps, I'll back up. If you look at this diagram here, you'll see this purple circles are the sodium. So, and the orange circles are potassium. Well, potassium in general is in a higher concentration on the inside of the cell. When the cell is just sitting there resting, it's not doing anything, there's more potassium inside the cell than outside the cell. There's more sodium outside of the cell than inside the cell. But look what the sodium-potassium pump does. It's pumping potassium in. It's pumping sodium out. And what that means is it's pumping against the concentration gradient. it has to pump from an area of low concentration to an area of high concentration. So it has to use energy to do that. It's moving 3 sodium for every 2 potassium. And it's doing that so that cell can maintain equilibrium with its environment. This sodium-potassium exchange pump is going to be hugely important when we talk about muscles, when we talk about nerves. So if you don't quite get it yet, sit it on the back burner, and when we get to that part of chapter 9, I think, that's when you're really going to have to make sure that you understand how this works. So there's secondary active transport. This happens a lot in the kidneys, where you have like sodium and glucose being co-transported in, and then that sodium's then being pumped right back out by the sodium-potassium pump. There's vesicles like endo and exocytosis actually forming little transport bubbles into or out of the cell. Endocytosis can be receptor mediated, meaning the cell can actually have little receptors on it that say, it's kind of like a landing strip, like land here, this is where you need to come in. And so those molecules will seek out that particular part of the cell membrane to come into the cell. Pinocytosis, phagocytosis, exocytosis are all forms of endocytosis, eating, drinking. Exocytosis is the reverse here. So you have this little bacteria coming in. This is endocytosis, and it's just forming a little transport bubble around it, and then all these little lysosomes from the Golgi sense that this is a foreign object, foreign particle. They come in, they engulf. Again, this is through endocytosis right here as well. Coming in, and then they engulf and just kind of destroy and dissolve that bacteria, and then exocytosis pushes out the remainder. These are just charts from your book summarizing what I just went through very, very quickly. Let's talk about transmembrane potential. Again, this is something that's going to come back to haunt you, so to speak, when we talk about muscles and nerves. It's the idea of a resting transmembrane potential, or RMP. For various cells, it's going to vary. It's going to vary from cell to cell, from cell type to cell type, I should say. It could be -10, it could be -100. It's usually a negative number, and the negative is just saying that there's more negative ions on the inside of the cell than on the outside of the cell. For muscle cells, it's around -70, I believe. So that transmembrane potential is determined by that unequal distribution of positive ions, sodium and potassium. The interior has a greater concentration of potassium. The outside has a greater concentration of sodium, like I said. But because potassium is a slightly larger molecule, that positive charge carries a bigger weight, so to speak. So at rest, the plasma membrane is relatively impermeable to sodium. It's generally freely permeable to potassium because it wants that potassium to come in. And so you have these two types of channels, passive and gated, or leaky and gated. And depending on what is going on in the cell, those channels may be open or closed. But again, we're going to talk about these more specifically when we get to muscles and nerve cells. So more potassium diffuses out of the cell than sodium coming into the cell, and so because of that, our cell is now more negative than the resting membrane potential. We call that polarized. And once it's polarized, then it can be activated, and it can carry a charge, and it can propagate a charge, and it can carry information, and that's when the magic happens for muscle cells and nerve cells. If too much potassium is left in the cell, it becomes too negative, and then it's hyperpolarizing. It gets too hard to contract. That sometimes happens with our heart cells. And again, we'll talk about all of this more specifically. I just wanted to introduce the idea of transmembrane potential to you because it is hugely important later on, and it was covered briefly in some of your prerequisite courses. So that sodium-potassium pump plays a huge role in those membrane potentials. So when that stimulus is applied, here's the stimulus. When that stimulus is applied, those channels open and allow sodium to come into the cell. So you see the sodium is going from, or the cell is going from -70 resting membrane potential all the way up, becoming more and more positive because all of that positive sodium is coming in. Then the sodium channels close and the potassium channels open, and all the potassium can then rush out of the cell, and the cell comes back to normal. And this whole process, you say, okay, well, why do we care about that? It's going to make more sense when we look at how nerve cells and muscle cells interact. So let's talk briefly about cell division. I think that's our last little review topic. Mitosis and meiosis. I like to say meiosis. I don't know. But if you say the word meiosis, it sounds so different from mitosis, you're not going to get them confused. So mitosis and meiosis. Mitosis is cell division. This is happening every day with all of our cells, except our nerve cells and like our fat cells and stuff like that. But the majority of our body cells are undergoing mitosis as we grow and repair. Meiosis only happens to our sex cells, the eggs and sperm. It happens to form them. So mitosis creates two identical daughter cells. They are completely the same, have the same genetics, look, walk, talk, and sound the same, right? Whereas with meiosis, we're creating 4 cells, and those 4 cells are all unique, right? All the eggs that females produce are all unique. All the sperm that males produce are all unique. And they, of course, these processes are very, very different. Meiosis has 2 sets of divisions. So there's meiosis 1 and meiosis 2. And meiosis also has this process called crossing over that occurs during prophase 1 of meiosis 1. And that is when those sister chromatids are going to actually synapse and exchange genetic information, kind of swap some genes around so that every gamete or every new cell that's produced is completely unique. That process does not occur during mitosis. So mitosis can also be the cause of cancer. Mitosis is the normal division of cells, whereas if one of those cells goes rogue and does not follow directions, maybe it grows uncontrollably or divides uncontrollably, then you have a cancer issue. So when those cancer cells can escape and get into the lymphatics or into the bloodstream, they can travel into another part of the body, and that's when you have metastasis, and that cancer has then spread to other areas. So these are just some of the factors that affect cell division. So if any of these factors are mutated or wrong or not functioning the way they should, there's a risk for cancer development in that particular area of the body. So that is, let me see if I can I switch back to me? How do I do that? I don't know, can you see me? Hide thumbnail, show me. I don't know how to see me again. Oh, right here, sorry. There we go, hi, I'm back. So hopefully this little video helped you, and this is my first time doing the screen share with me in it, and next time we'll be flipping back and forth more. This was just a straight review, so this is probably one of the most boring PowerPoint lectures you'll see from me, and it's really just to get through the material. So hopefully this lecture helped you. If you're overwhelmed, here are some things you can do. If I went through that and you felt like I was speaking Greek to you, one of the things you should do is go back to the chapters, read them, use the reading guide, and hopefully that will fill in some of the blanks. If not, then you might want to consider withdrawing from the course early and taking those prerequisite courses. I know sometimes advising lets students in that haven't had a bio class in a very long time. In some cases, they're a little more strict. So use your best judgment. If you think you can handle it, if you think that you can master that content, awesome. Do you need to know every bit of information about lipid in order to be successful in A&P? Absolutely not. But you should know the basics. You really should be familiar with what I just talked about, because all of those things are going to come back and hit us in the head this semester and next semester in A&P too. So it's super important that you are the master of this material so that we can move forward. So good luck to you, and please let me know if you need help.